Morning, everybody. Thank you, Christine, for that introduction. I'm Gemma Carvill from Northwestern, my colleague Heather Mefford from St. Jude. We're going to do things a little different in that we're going to tag team today in our opening presentation. Can everyone hear me at the back? Yeah, great, cool. So super excited to be here. I think this is an amazing turnout. Before we even get started, I want to say a huge thank you and congratulations to Christine and Lauren. It's truly remarkable what you guys have been able to do in such a short period of time. It's very exciting to have everybody here today. I think 50 families is really incredible for a first meeting. And also a big shout out, of course, to Christina and Stephanie for helping put all of this together. It's been a real tour de force, and you guys should all give a big thank you to all of the above. There we go. I agree. And I think it is even more impressive that we're all here today, given the very short period of time, relatively speaking, that Christine and Lauren have managed to pull us all together into this room. CCC was really only started back in 2020, so less than 3 years ago. So this is an amazing feat. We put this timeline up here, really outlines our path to this point. We're sitting here in 2023, and I'm sure in 2020, Christine and Lauren did not quite believe that in 2.5 short years that we'd all be sitting here we will host the first ever family conference and research symposium, and here we are today. So I think this is an awesome feat. Super excited to be here. And so I thought what we'd do to start today is I'm going to open with giving you a brief overview of CHD2, how we identified the gene, and just get us all on the same page in terms of some basics about this gene, the reason that we're all here. And so we first identified pathogenic variants or mutations in the CHD2 gene back in 2013, which is wild to think. I was writing this down the other day. It's 10 years ago that we discovered this gene. And this was really first identified in individuals with epileptic encephalopathies. So there's very severe types of epilepsies. And this very first paper back in 2013 was just 5 individuals. And so as you guys all recognize, this has really grown in the last 10 years in terms of the number of individuals. But the other thing that we know about this condition is that it's evolved over time in terms of the symptoms that are associated with CHD2 mutation. And so now we know that seizures, developmental delays, and behavior are very different from individual to individual. And it's a lot broader and variable than we first described. The other hallmark of CHD2 is that variants tend to arise de novo. And so what I mean by that is everybody in this room has 2 copies of the CHD2 gene. And what occurs in the individual who is affected with CHD2 is that they have a mutation in this gene that they in general did not inherit from either of their parents. And so we call this a de novo mutation. And so in most cases of CHD2, this is the scenarios where it tends not to be inherited from either parent. So getting back to how many patients are there, we described those first 5, 10 years ago. We've collected genetic data from a variety of different resources from over 200 individuals. There are 50 families in this room, which means that this is almost certainly an underestimate. We really don't know how many individuals are out there. And so things like the Facebook group, all of the work that CCC does in terms of bringing everybody together is really helpful in shaping how we find a cure for this disorder. We do know that it is one of the most common causes of neurodevelopmental disorders. And I'm sure many of you have enrolled in RareX. So this is an online clinical and genetic data repository, essentially. And to date, we know that there are about 108 individuals enrolled in RareX, and we are distributed across the world, as you can tell. One of the other real characteristics of CHD2 mutations or variants is that they tend to lead to loss of that protein. And so just to take you through what I mean by that, I have a schematic here that shows the DNA, or an expert of the DNA, the codes for the CHD2 gene. And so DNA is then made into RNA, and this RNA is then translated into a protein. And a protein is just essentially a string of what we call amino acids. And so I'm showing you here 5 amino acids. There are actually 1,800 that make the CHD2 protein. And what characterizes all proteins in our cells is that at the very end of those that string of 1,800 amino acids is you have a stop codon at the end. That's how you know you've got to the end of the protein. And so what occurs in individuals with CHD2, in general, the vast majority of individuals have what we call a stop mutation. And so here, I'm showing you in red that this is the mutation in red here. And so you have the DNA, makes the RNA, and now what ends up happening is that there is a new stop mutation much earlier in the protein. And that triggers a whole host of processes in the cell, which then degrades the protein. And so this is what we call a truncating variant. It means that it does not make a functional protein. And then some individuals will have a missense variant. And so in this example, a different mutation in the DNA sequence is coded into the RNA. And now in the protein, you can see that this purple amino acid has simply switched to a green one. And if this occurs at a very important part of the protein, it tends to render it non-functional. And so an individual still has 2 copies of CHD2 protein, but one of them is non-functional. And so essentially what happens in these 2 instances is something that we call haploinsufficiency. And all this means is that the individual with a CHD2 mutation only has 50% of functional protein as compared to most individuals in the population. And having only 50% is therefore then associated with the symptoms that we see in patients with CHD2 mutation. One of the other big questions is, what does CHD2 do? And how does it lead to the clinical symptoms that we see in patients? So CHD2 is responsible for folding how the DNA is folded into the cell. So if you had to take a DNA sequence from a single cell and wind it out in a long twine, essentially, it would reach many, many different football fields. It's that long. And so, but it somehow has to be packaged into our cells, billions of cells in our body. And the way in which that happens is it essentially folds up into a tiny little ball. And essentially what CHD2 does is controls how tightly wound that DNA is. And so if it's very tightly wound, any genes in that region tend not to be expressed when they're not switched on. So that's what I'm showing you. DNA is very tightly wound, and a gene can't be expressed. The job of CHD2 is really just to open up that DNA, and so the gene can be expressed, and you can get protein. And so CHD2 does this with lots and lots of genes in the genome. And really, many of these genes tend to be involved in brain development function. And so when CHD2 is not there, this opening up tends to not happen, and then you get changes in gene expression. And some of the work that we're doing on the research side is to figure out, what are these genes? What are the pathways in the cell that are affected that then leads to the symptoms that we see in patients? And so what Heather and I are going to do today in terms of our presentation is we're going to outline the roadmap to a cure. And so this is a roadmap that was developed together with CCC to give us a schematic of how we think about all of the steps that we need to take towards treatments and cures for CHD2. And so at this point, Heather and I are going to tag team and take you through all of the steps that we need to take along this road. And so Heather's going to lead off with identifying and defining the patients with CHD2.
All right, great. So with that introduction as to what is CHD2 and what does it do, we now need to think about how do we identify all the patients who are affected, how do we move through a both scientific and clinical research process to get to a better treatment or even a cure? How do we identify the patients? Well, you are all here because you already have, or your child already has, a genetic diagnosis. So in some ways, you're lucky to have that. There are a lot of people who have not been able to access genetic testing. And so one of the questions that we need to think about is how do we increase the number of families and individuals in this room and in research projects so that we can better understand CHD2, get them all involved, and give them access to new therapies that might be coming down the road. So most of you have probably gone through a series of genetic tests, I'm guessing. Some people have probably been through many. Some of you may have had a diagnosis with the first genetic test that was done. But in the clinical space, whether you're seeing a neurologist or a geneticist or a developmental pediatrician, we have a wide array of genetic tests that are available. So some of these you may recognize. Often kids who have developmental issues early in life, the first test they get is a chromosome microarray. So this looks for little pieces of DNA that are missing from our genome, or they are in an extra copy. But more and more today, the first tests that are done or that should be done are sequencing tests. So either gene panels that look at that DNA sequence or the spelling of the DNA across many genes or all genes, or even whole exome sequencing, which some of you may have had, which looks at every single gene and tries to find the single spelling error that is the cause of disease. But we have more than that. There are many different tests now that are coming online. I'll talk about one of them in a little bit, but we can look at not just the DNA sequence, but we can look at the RNA sequence. We can look at additions, little modifications to the DNA called methylation, which I'll talk about in a minute. And I'm not going to talk about the last one, which is right now does not pertain to CHD2, but some other types of epilepsy may actually be caused by small changes in many genes, not just one gene. And so one of the challenges, just so you know, from the clinician's perspective, is to decide which of these tests do you do first and for which patient, right? And so we need to, one of our jobs, our jobs, and some of the physicians in the audience is to educate all of the clinicians out there to say, these are the tests that you should do first when a child or a family comes to you with concerns about their child with developmental delays. Do this test, right? And I'm guessing that some of you in this room had frustrations with saying, there's something going on that's not right, and it took a long time to get that first test, right? So we're working hard to try and improve that situation. That said, even when we can, educate the clinicians, there are often other barriers. But in the very recently, in the past year, there are now recommendations from the National Society of Genetic Counselors for kids who come in who present with epilepsy specifically, or any individual who has epilepsy, that the first test they should have done is a whole exome sequencing test, which is kind of the best test we have to offer today. And if we can't get that, they should at least have a gene panel which looks at multiple genes at once. And then that should include counseling from a genetic counselor about what you might find or might not find from some of these tests. So this is a recommendation that's in the literature, and it's been endorsed by the American Epilepsy Society, which means every physician should be thinking about this, but we know that it still doesn't happen. And I know some of the barriers, one of the big barrier is insurance coverage, right? But we need these kind of recommendations from respected medical bodies to say to the insurance companies, you should cover this, and this is why. And just so that you all know, there are some activities or efforts underway to try and improve this nationwide. So this is specifically for the United States. So we have many recommendations now, not just the one I just showed you, but also recommendations that kids with not epilepsy but developmental delays should have genetic testing, and that first test should be an exome sequencing test. But again, getting it covered is hard. But there are at least 2 bills that are working their way through the governmental process, which is complicated in itself, as we all know. But one is called Ending the Diagnostic Odyssey Act, and the other is an Access to Genetic Counselor Services Act, which will give us more. One will give hopefully encourage insurance coverage for things like exome and genome sequencing across all patients. And the other will give access to allowing genetic counselors to actually order tests. They are as knowledgeable as the geneticists in this room and can recommend this testing. So I encourage you to take a look at these and think about as a rare disease group, joining forces with other rare disease groups, right, to contact your senators or congressmen and congresswomen and encourage them to pass these bills. If you get access to genetic testing, and there are many different results that you can get, and I just want to bring this up because I think some of you have probably been through this and asking what does pathogenic mean? What does benign mean? What does variant of uncertain significance mean? So when you get a genetic test, you can get several different, if they find a genetic change in you or in your child, it can be interpreted in different ways. And what we're looking for when we do a genetic test is a pathogenic change. And pathogenic really means disease causing or damaging. But there's a wide range of what you can get. You can get a result that's likely pathogenic. That means we think this is causing your disease. We're pretty sure. The difficult interpretation to get is a variant of uncertain significance. This means we found a genetic change, but we don't actually know if it's causing the issues that you see in your child, or if it's actually just a genetic change that we all have genetic changes in this room that don't actually cause any problems. And then benign changes are just changes that, again, that we all have that have no effect. That's how we interpret the effect of a genetic change. And then the test result might say, this is, if you have a pathogenic or likely pathogenic change, we interpret that, we being the physicians or the geneticists, and interpret that as a positive test. So this means this is diagnostic, this is the cause, this is your genetic disorder. A benign variant doesn't have any effect, that's a negative test. And I would say, and there may be some of you in this room with variants of uncertain significance, so we're happy to talk to you about that. But a variant of uncertain clinical significance is actually not positive diagnosis. It's a change that needs more investigation and more interpretation by experts in the field. And often, when we do a little bit of extra work, we can say this is a positive or a pathogenic change. And on the other hand, there are many times where we do a little bit more work and we say, actually, this is not the cause and we should keep looking. So these are, and as we acquire information over time, the number of variants of uncertain significance that we find goes down because we gather more and more information about changes in every gene in both affected and unaffected individuals. But what if you do have a variant of uncertain significance? What's the extra work that we do? Some of that extra work is talking to people who are experts in understanding the gene and those specific changes and whether they might be affecting the protein or not. But today there's actually additional clinical testing that you can do to ask, is my variant of uncertain significance a positive test or a negative test? And one important test that has emerged over the past couple of years is something called EpiSign. This is a test that doesn't look at the DNA sequence of CHD2 or the spelling of CHD2, but it looks at other changes in the genome that CHD2 might be affecting. And those are changes called methylation. So methylation are little additions to the DNA sequence. And we know that, as Gemma said, one of the jobs of CHD2 is to unwind that DNA, and it's part of that process is through methylation. And kids who have CHD2 pathogenic or disease-causing changes have a specific methylation pattern in their DNA that's different from kids who don't have CHD2. So if you have a variant of uncertain significance and you do the EpiSign test and it has the CHD2 methylation pattern, that helps us a lot to say, okay, this is definitely a change in CHD2 that's probably causing the disorder in this child. And if they don't have that change, then we can go back and say, okay, what else do we need to look for and how do we interpret this? So outside of the EpiSign test, as I said, you can talk to your genetic counselor or your neurologist or your geneticist. And we're here today. So I know many of you met Emily Bonkowski on the way in. She's a genetic counselor with us at St. Jude. If you have questions about your genetic test report and you have it with you today, we'd be happy to talk to you about that. Even if it's a clearly pathogenic change, if you want more information about what that means, we're happy to talk to you. And you can find her. She's right back there wearing her CCC t-shirt, and we'll be around today and tomorrow. Okay, so what next? You have a genetic diagnosis. What do we need to do next to gather information to understand the disease and to use that information then as we move towards having better treatments and clinical trials to use the clinical outcomes for to understand if a new treatment is working. So many of you in this room have probably enrolled with RareX, as Gemma already mentioned. Some of you may have enrolled in another program called Citizen or Invitae. But these are two different ways for us to gather clinical information, either from you filling out surveys about your child in the RareX case, or gathering clinical information from your medical records in the Citizen Invitae case. These are both different than us talking to you and gathering information directly from you, which we hope to do for many of you over the next day and a half. All of these are important and collect, I think, slightly different types of information that can be interpreted in different ways. But I think it's important to think about enrolling in these so that we gather as much information as we can about your children. As I said, some of you enrolled on the way in. We're very excited about having you involved in our research program so that we can take not only the information that you give us here and as we follow up with you over time, but ask questions about how do we compare what the data that RareX collects to the data that Citizen collects, to the data that we get from you by talking to you, and to the data that some of you have probably talked to Christina, who's interviewed some of the families here about what are the most important outcomes that you're looking for your children, and integrate all that data to then say, okay, what's the most important thing for us to focus on as we develop new treatments? What is it that's most important to you that improves for your child, right? Or that we cure for your child? And I think sometimes as physicians, we have our own ideas about what's important or interesting, but we need to coordinate with you and say, we need to meet your needs about what's most important for us to affect in your child's development or long-term health. And then I think what's really exciting is yesterday we had an afternoon of researchers and clinicians presenting the most up-to-date research that we're working on. And some of that was about the clinical features of CHD2. So Dr. McTagg is in the UK and talked to us about sunflower syndrome, which is a photosensitive epilepsy for which many of those individuals have CHD2 mutations, though not all. So there are multiple causes of that. You're going to hear from Dr. David Lewis-Smith, who's here today and talked to us yesterday, but we'll also talk this afternoon, I think, about some of the clinical features and how we extract information from the medical record. We have Dr. Andrade from Canada, who's an adult neurologist and is doing excellent work in studying some of the disorders that we identify mostly in children, but those kids grow up, right? And so adult neurologists need to understand, what does it look like when a child diagnosed with CHD2 at 2 years old, when they're 22 or 42, what does that look like and how do we best care for them? And then Dr. Kelly Knupp is here from Colorado and is going to talk to us this afternoon from a pediatric neurologist point of view. What are some of the important things to understand and to focus on as we move forward? Okay, and I think I'm going to tag team again. So that's thinking about the patients, identifying the patients, defining the clinical picture. And now what do we do next as we move from the patient back into the lab to understand CHD2, and then we'll come back to the patient again at the end.
All right, so next step along our roadmap to a cure is thinking about models of CHD2, and then using those models to study CHD2 and to find new therapies. And so there are now a variety of different models at our disposal that we can use to study this disorder. And to start with, I'll talk about animal models. There are a variety of different animals. So I've highlighted here, mouse models are very commonly used to study the genetic epilepsies and all rare and common disease. We can also use zebrafish. So zebrafish are tiny little translucent fish that are also actually pretty good models for genetic disorders. And then lastly, more recently, actually, folks have started to use frogs for studying particularly neurodevelopmental disorders as well. And so what we do with all of these different animal models is that we can knock out 1 or 2 copies of CHD2. And all knocking out means is, as the name would imply, that you just delete the gene and make sure that there is either 50% functional protein or no functional protein at all. And because CHD2 is highly conserved throughout evolution, CHD2 can have similar roles within the cell, even though humans and these species are very different. Knocking this gene out can tell us a lot about the biology of CHD2. And so what we do with these animal models is we study the role of CHD2 in the cell. And so when we knock it out, what are the genes whose expression changes? What are the biological pathways? If we look in the brains of these animals, how are they different? Do they have a slightly different morphology? Do they have a slightly different shape? We can examine all of these things within an animal model. We can also look at things like, if we knock this gene out, do the animal models exhibit seizure behaviors or seizure-like behaviors? In this instance, it's a great model because, as all of you in the room know, seizures is really one of the defining characteristics of this disorder. We can also study things like behavior, particularly in mice. We can study learning and memory, again, more in mice, but a little bit in the other models as well. And once we have established what we call the phenotypes in these animals, we can then start thinking about testing new therapies. So we've understood the biology, we have an idea about the pathways that are affected, and we have something that we can measure. And once we're there, we can then think about what are the therapies and see if we can reverse those phenotypes that we see in the animals. And so yesterday we had the research symposium, and many folks within the research community are working on CHD2 and working on a variety of these different models. And so I've just highlighted here some of the researchers that are really focused on this particular disorder. And I'm sure we're also going to hear about many of these today. I think this afternoon and into tomorrow, we're going to hear about some of these frog models, as well as the mouse models that are being used to understand CHD2 and the biology. In addition to animal models, we can also use what we call cellular models. And so this can mean a lot of different things. There are many, many different types in the body, different types of cells rather in the body. But by and large, what the vast majority of the scientific community is doing is using iPSCs or stem cells. And so just to take you through that process, how we go about using these cellular models, we start with a patient with a CHD2 variant, and we can either take a very tiny skin sample from that individual, or we can take a blood sample. We then isolate the cells from that blood draw or the skin sample, and using a very nifty method, we can actually what we call reprogram them. And so we take those cells back to a state where they can become, using a variety of different compounds, in theory, any cell in the body. And so we take these stem cells, and because we're primarily interested in the neuronal phenotypes or the brain-specific phenotypes of CHD2, we take these stem cells and we differentiate them into neurons. So these are the major cells within the brain that are responsible for communication and signaling, if you will, in the brain. So we can differentiate them in this tiny little dish into what looks like neurons. Many of us are also using organoids. You may have heard the term mini brain. And these are essentially 3D structures that have many of the cell types and recapitulate a lot of the way in which the brain develops in an actual little dish. And so these are a more recent technology, again, hugely powerful in terms of studying how CHD2 mutations affect not only how the brain develops, but also potentially how it functions. And so very much like the animal models, we can also study the role of CHD2 in the cell. So earlier I said how the animal models are great in terms of, because of evolution, the biology is likely highly conserved. But we all know humans are not tiny mice, they're not tiny zebrafish. And so one of the real advantage of this particular technology is that CHD2 might have human-specific functions, that really using these stem cell neuronal protocols allow us to examine the potential human-specific characteristics of CHD2. While something in a dish cannot have a seizure, it can have seizure-like activity. And so we can measure things like electrical currents, we can measure how these cells in the dish communicate with one another, and we can compare those individuals with CHD2 mutations to those that do not, and we can see if there is a difference in how these neurons communicate in the dish. And so we can sometimes see activity that looks like seizures. And so again, that gives us a nice platform to in the future begin to test new therapies in this model as well. And so again, yesterday, there were a number of researchers that spoke about their stem cell technology work, my lab included, and I'm very happy in the breaks, over lunch, dinner, to talk more about the work that we're doing in my lab. But there are also labs across the world working on this. And so Neil and Ellie are in Nijmegen in the Netherlands. And Ellie gave a lovely presentation about those neurons. So these are just the 2D version. And she's specifically looking at how these neurons communicate in the dish and whether we can see seizure-like activity. And her initial results are very encouraging in terms of there being differences between individuals with CHD2 and those without. Jenny Shea is a researcher at UTSA, and her and Vanessa are working on organoids. So again, those are the mini brain, the 3D version of brain development. And they also have some exciting work on looking at the types of neurons that those organoids make, and are they different in CHD2 patients versus not. And then lastly, Kristen Kroll at WashU is focused on a very specific type of neuron in the brain. And so the neurons in the brain, there are over 200 types. There's probably more that we don't even know about. And GABAergic interneurons are just a very specific type of neuron in the brain that has a specific function. And we think CHD2 is very important in the development of this specific neuronal cell type. And so that's the work that Kristen is working on in her lab. And so now we have these models, and we're not all the way there in terms of figuring out the biology and having good phenotypes in both the dish and the animal model. But it seems strange to say, considering we only identified the gene 10 years ago, this is pretty remarkable progress in terms of having these models and starting to understand. And so what we'll talk about next, and we'll talk about now that we have these models, how do we move towards testing therapies in these models?
Great. So this is actually
like do full screen and all that.
Somebody's not on mute. If you're online, make sure that you're on mute, please. Great. So I think this is a really exciting time in genetics right now to think about how do we develop new treatments. So as an example, many of your children may be on anti-seizure medications that may or may not work, or to varying degrees. Many of those medications are not specific for CHD2 epilepsy versus SCN1A epilepsy or gene X, Y, or Z epilepsy. But some of the tools that are emerging today are allowing us to say, okay, now we know the genetic mutation. Is there a way that we can actually be more specific about going in to treat that specific genetic mutation or that specific gene cause? And so as Gemma already nicely introduced for you, CHD2 is a disease of what we call haploinsufficiency, which basically means you don't have enough working CHD2 protein. So a simple way to think about this is those of us in this room who have 2 working copies, that's great. We don't have any seizures or developmental issues. Those of us in this room who have only half of the amount of CHD2 are affected. So is there a way that we can just increase the amount of that working copy that's left, right? Because kids with CHD2 have 1 copy that works and 1 that doesn't. Can we increase that enough to actually have an effect, or will that have an effect, right? So that's just shown here. And as we all know, CHD2 is caused by that single mutation. CHD2 disease is caused by that single mutation in the DNA. So how can we go in and ask, can we fix that or affect it to ameliorate some of the effects? What's really exciting, as I said, in the field right now, and this is true not just for CHD2, but for many of the other genetic epilepsies that some of us study, is that there are tools, and you may have heard the term gene therapy, CRISPR, genetic therapy. All of these are becoming a reality, and they are a reality for some disorders. And just some examples of types of therapies that you might hear about. One is called antisense oligonucleotide therapy, or ASO therapy. These are tiny little pieces of RNA that you can match a piece of DNA to go in and either turn up the amount of the gene that's expressed, turn down the amount of gene that's expressed, or have some other effects. SA or small activating RNAs are very similar in that way, in that they're small pieces of RNA that you can go in. And the nice thing about designing these therapies is that you can say, go to CHD2 and have an effect, or go to gene X and have an effect, which is different than many of the medications that are used today. Many of you may have heard about chaser. So chaser is a RNA that all of us carry, a piece of RNA that actually helps turn on or turn off CHD2. And so we think that this is something that may be a target, not targeting CHD2, but targeting chaser, which helps increase the amount of CHD2 that is made. So these are ways to go in, and I like to think of it as turning up the volume or turning down the volume of a specific gene, right? There are also ways you can just replace that copy that's not working. So kind of true gene replacement is where you give a new copy of the gene, it finds the cells, it integrates into the DNA, and now you have an extra working copy of the gene. So this is actually just replacing a non-working copy. If you've heard about CRISPR, CRISPR is a way to edit the DNA or change the DNA sequence. This is a little harder to think about as an actual therapy for something like CHD2, where you have to go in and change the DNA sequence in the brain. It's a little bit scary and it's a little bit hard to do. But it is a technology that's being used for some disorders where you can actually take out the cells or the stem cells, fix the genetic mutation, put them back into the patient, and then you have your working copy of the gene. CRISPR can be used in other ways, and one of those ways is actually, again, to either turn up or turn down the volume, or turn up or down the amount of protein that's being made. I think one of the important things we need to think about is that we know that changes in CHD2 mean that there's not enough CHD2 around to do its job, and that's what's causing the disorder. But there are individuals who have changes in the chain. To approve by the FDA to approve a gene therapy or gene replacement for Duchenne muscular dystrophy. Duchenne is another neuromuscular disorder that primarily affects boys, but it's one of the most common of those. And so that's in the process of having going through clinical trials and being approved for an actual treatment for Duchenne muscular dystrophy. And sickle cell anemia is a disorder that, again, we've known about for many, many decades. And it's a single gene disorder that causes a blood disorder. There's now a CRISPR type therapy where you can, because it's a blood disorder, blood is easy to access, right? You can take those cells out, you can fix the mutation and put them back into the patient. Actually have an effect in treating the disease. So there are many more examples, and some of the SPG is another neuromuscular disease that now has a gene therapy that's available. There are many metabolic diseases that also have gene replacement or enzyme replacement therapy. So this is becoming a reality. It's not yet a reality for CHD2, but this is what many of the individuals in this room and online yesterday and in the scientific community are thinking about, is how do we use some of these therapies specifically for CHD2. And as I've already alluded to, I think there's a lot of excitement, and I think we also have to be realistic and say, okay, let's remember to think about we don't want to cause harm with our therapies, and so how do we figure out what's the right amount of CHD2 healthy, not too much, not too little. How do we get our therapies to the brain, right? The brain is a very protected organ, and not all drugs get to the brain. So in addition to just designing the therapies, there are many people working on how do we deliver the therapies and get it across that, what we call the blood-brain barrier, and into the right cells that it has an effect, to effect on seizures and possibly development. How early do we need to treat kids with CHD2? How late can we treat individuals with CHD2, and what effect will that have on development, on seizures, on other conditions? And then importantly, I think thinking about if we give a treatment and there's a side effect, can we stop that treatment? And when you go in and replace a gene in the DNA or fix the DNA, you can't take that away. But many of these other therapies you can treat, and if there's a side effect, you can stop the therapy, and hopefully that goes away. So these are all things that I think are important considerations as we move forward. The field is moving extremely fast. So, Gemma said we identified this gene 10 years ago, and that may seem like a long time. Why the heck don't we have therapy yet? We identified sickle cell decades ago, right? And now the first therapy is available. But I don't think it will take that same amount of time for many of the diseases that we identify today, because we have these new tools and science is moving faster and improving quickly. Okay, so finally now we're thinking about all these things that we've gone from the patient and identifying the patients, thinking about how do we develop models and test new therapies, and now how do we come back to the patients. We've already said we'd love to have you all involved in research in some way, and this can be collecting information, talking to us, sending us your medical records. It can also be providing samples for us. So the cell therapies or the cellular models that Gemma talked about, which use stem cells from patients, require material from patients. And often that's just a blood draw or a skin biopsy. And so we're working, as we enroll you, we'll follow up with you once you get home to do remote blood draws, either in your clinic or in your home, to be able to collect some of that material. We probably don't need it from everybody, right? We need enough examples of the types of mutations that are important, the kids with seizures or without seizures, for example, to study these in a dish. We probably don't need to do it for everyone, but we want a nice representation of the populations that we can study this. Aside from making cellular models, we can also use DNA from blood, for example, to study that methylation pattern that I told you about that we can use. That CHD2 kids are different than other kids. And what we don't understand is why is that methylation pattern different in CHD2 kids, and how does that relate to disease? And is there something that we can learn about CHD2's effect in the cells at changing that methylation pattern and what that means for other things we might be able to fix or to treat? And then there are many efforts in the field to look at other types of biomarkers. So biomarker can be many different things. But you may hear this term a lot. A biomarker is something that we can identify, that we can quantify, so that we understand it in kids with CHD2, and hopefully we change it when we treat effectively, right? So that might be an EEG biomarker. So what does their EEG look like, and if we treat, can we change that back closer to normal? And a biomarker can be something in the blood that we can detect and test, like I said, identify, that when we treat kids with CHD2, that changes back to normal. So biomarker is something that we can quantify or identify that's different in kids who are affected and kids that are not. Well, we're going to start with primarily blood or skin, like Gemma said. These are very, very useful and accessible tissues. Down the road, we may require other types of tissues. CHD2 is a brain disease, The fluid around the brain is the cerebrospinal fluid. It's the closest to the brain, and the changes in biomarkers that are most important for kids with CHD2 might not be in the blood, or might not be in the skin, or might not be in the urine. They may be in tissues that are a little bit harder to access. So we'll learn that as we go, and learn what best. Is that for me personally? Hit on you. So as we move forward, we need to think about what are the tissues or samples that we need to collect and why, and what are we going to learn from that. So the other thing I will say is that, we've talked about, many of you, about enrolling in research. It does not require you to provide blood samples or skin samples. It's just something that we think can be useful in our research, and we would encourage you, if you're willing, to think about being able to do that, but it's not a requirement. We're still going to learn from all of you about the clinical picture of CHD2, and then how we can change that down the road in clinical trials. And then finally, what do we need to do moving forward? You've heard about all the exciting things that we're hearing about, learning about, and that we are researching about CHD2. But what are the next steps that we need to do to actually get to a better treatment or a cure?
So to round out our journey along the road, what do we need to do together? So many of the topics Heather and I have gone over today along that roadmap, we're going to talk about in more detail today and tomorrow. In the research symposium yesterday, we spoke about many of the scientific efforts that are ongoing, and we identified specific challenges and opportunities. And so tomorrow in closing, I'll talk about some of those as well. But most of all, we want to hear from you. So we want to hear, what questions do you have and what's important to you? And so to start to get a sense for what is important to the CHD2 community, Stephanie Prince, one of the board members, did an initial poll on the Facebook group to get an idea of what are family priorities. And so she polled the Facebook book group for the top 3 research priorities via email. She then created a poll, and everyone could vote for their list of top 3 priorities. And what I'm showing you here is a graph of what you guys in the Facebook group told us are your biggest priorities in terms of your kids with CHD2. And so what you'll see here is that seizures, behavioral issues, as well as developmental delay and intellectual disability are really up there in terms of your biggest priorities. And so what I'm showing you is just from the initial poll, but this pattern very much held when we polled the larger group, and there were 75 respondents. And so this is great because this gives us an idea of what's important to you, but we want to go into more detail. And so the slide is also sort of showing you what some specific questions that the community asked. And so you can read some of them, not all of them, but we want to be able to add to this list. And so we'd love to know, what else do you guys want to know? We have a day and a half here. We have a fantastic community. We have a lot of scientists. We have a lot of physicians here, and we really want to know what's important to you and what questions do you have. So come find our teams in the break. So I'm showing all of their photos here. So both the team, Heather's team from St. Jude, as well as my team from Northwestern. Find them in the breaks, at dinner, ask us whatever you want to know. And then the goal will be that we'll all regroup and we'll kind of come up with the top 4 or 5 questions that you guys have as a community. And then in closing tomorrow, we'll see which of those questions we can address. I can unfortunately guarantee you that we're not going to have all of the answers, but we'll be able to tell you where we are and the steps that we're taking in order to address your questions. And so with that, I will end. Thank you very much for your attention. Very much looking forward to the rest of the meeting. Thanks. Do you want us to take questions or no? Perfect, great.
Thank you, Gemma and Heather. Fantastic job giving us the roadmap of not only what we're going to try to accomplish in this conference, but really what Coalition to Cure CHD2 was designed to do, which is to help advance research to ultimately an effective treatment or therapy for our kiddos. So fantastic way to tie that all in.